So welcome. Welcome to our center's Future Leaders in Mechanobiology Seminar Series. And welcome, I'm happy to introduce Liana Boraas today. She's our speaker, and she's currently a postdoc at Yale University in Dr. Nicoli's lab. And she received her PhD in biomedical engineering, and her PhD advisor was Dr. Tabi Hassan. And she received her bachelor's degree in engineering from Harvey Mudd College. So we welcome Liana today, and go ahead and share your.
Great. Yes, thank you so much for that wonderful introduction. I'm Liana, and I'm very excited to be giving the seminar series today all about untranslated mRNA sequences and how they regulate cell movement via a non-coding function. So Trina talked about this, but I do want to just briefly highlight my educational background because I do feel like it shapes how I think about biological problems, especially now having come from an engineering background to the School of Medicine at Yale University. So I did my undergraduate at Harvey Mudd, biomedical engineering PhD at Tulane. And as I mentioned, now I'm at Yale as part of the genetics and internal medicine departments. And so when I was an undergraduate, I took a lot of systems and signals engineering. So when I was doing my PhD in stem cell mechanobiology, I was really captivated by this concept of the cell as a transfer function. So I'm sure all of you are familiar, but the idea that we can model systems using a transfer function that essentially governs the way that systems respond to particular inputs. So in this case, when we think about a cell, there are different microenvironmental signals, such as biomaterials or soluble factors. And then depending on the cell, what proteins it's expressing, it can interpret those microenvironmental signals to then create an integrated response. So if we want to engineer a particular cell response for therapeutic purposes, we can either change those microenvironmental signals, or we can actually augment the biological transfer function itself, assuming that we understand it, in order to get the integrated response that we want. And this is particularly valuable, especially in situations where we actually can't change those microenvironmental signals. So let's dive into the cellular transfer function and think about one, if not the most fundamental component, which is the central dogma, the idea that DNA gets transcribed into mRNA, which gets translated into protein. Now, it's unfortunately not that simple. There's regulation that goes on at a number of different levels. My lab primarily focuses on mRNA post-transcriptional regulation. So there are a number of examples of this. In the nucleus, after mRNA is made, there's 3' cleavage and polyadenylation, as well as RNA base editing. And these change ultimately how the protein is made when mRNA is then shuttled into the cytoplasm. In the cytoplasm itself, mRNAs can also be regulated by things like microRNAs, and RNA can also be localized to particular subcellular compartments where it may function there. So I don't have time to go into this work today, but I would like to highlight the work that I did when I first joined the lab, which was a story all about microRNAs and how they regulated the cell response to different substrate stiffnesses. And so this was a work that was done in collaboration with Martin Schwartz's lab, and we looked using both cell culture and zebrafish model systems to understand how microRNAs may regulate tissue homeostasis. So that work is out in Nature Cell Biology. Today, instead, I'm going to focus on RNA localization, and especially its role in mechanobiology. My lab studies post-transcriptional regulation. I have an interest in mechanobiology, so I wanted to merge these two fields for my postdoctoral work. The original idea actually started in the late 90s in Don Ingber's group, where they had taken beads that were coated in integrins, and they had allowed them to adhere to cells, which when this occurred, they saw a localization of polyadenylated mRNA as well as ribosomes. This work then continued in Bob Singer's lab. He was actually an author on that original Nature manuscript, and he looked at migrating cells, particularly migrating fibroblasts, and saw that beta-actin mRNA localized to the leading edge of these migrating cells. So now there are a handful of other groups that are really starting to dig into this concept of cytosolic mRNA localization and how it might be involved in mechanotransduction and cell migration. Here's an example from endothelial cells, both in 2D and zebrafish, as well as in 3D spheroids and mouse models in cancer. Where do I fit in? Well, since my graduate work, I've always been fascinated by the cytoskeleton and focal adhesions. And this seemed like a particularly rich candidate because, as I'm sure many of you are familiar, focal adhesions are highly localized structures. So we're talking about RNA localization, and now we have these highly localized structures that are responsible for connecting the cell to the extracellular matrix. And they facilitate how cells interface with the ECM, as well as facilitating things like cell migration. And so the idea was that, okay, is there RNA localized to focal adhesions? And if so, could it be involved in mechanotransduction and mechanobiology? So that is really what spurred the motivating question for my project, which was, how do these localized cytosolic mRNAs play a role in mechanotransduction? Okay. Now, in order to do this, we first needed to identify what RNA, well, I guess if RNA was present at the focal adhesions, and if so, what were those RNA species? So that's the very first experiment that we did. In the lab, I took the technique from Claire Waterman's group, where cells are cultured on a flat substrate, treated with a low ionic strength buffer, and then using hydrodynamic force, we can remove the cell body, leaving only the focal adhesions as well as some of the remaining cytoskeleton. And so with this approach, you can see we can identify the focal adhesions still attached to the substrate, as well as some of the actin that was attached. It's impossible to remove it all. You'll end up removing the focal adhesions without it. But when we did a Western blot of this, we can see that we removed the majority of negative control proteins, cytoskeletal proteins, as well as ER and mitochondria, while retaining our focal adhesion proteins. All right, so with this assay, we then wanted to look at what RNA is there. So we did poly-A RNA sequencing. We did ribosome profiling to determine whether or not the RNA was translating. And we did proteomics to determine if it was contributing to the local proteome. So with these assays, we took isolated focal adhesions and compared them to whole cell controls. We did this for endothelial cells, HUVECs, as well as fibroblasts. So these are both primary cell lines. And when we did this, we identified populations of mRNAs that were localized to the focal adhesions. And this was true for both cell types. We can then overlay the ribosome profiling and proteomics data to determine if those RNAs are translating. And what was extremely surprising to us is that actually the majority of these focal adhesion mRNAs were not translationally active. I think it was like 83 to 87% of the mRNAs were not translating. This was surprising to us, as the prevalent theory in the field is that localized mRNAs, of course, they're there to locally make protein. So we wanted to validate that these results were correct. For this, we turned to two different imaging-based approaches. The first is single molecule FISH. That allows us to identify the mRNA that is present, and we can identify one mRNA at a time. So here are two different examples, one in the untranslated pool, one in the translated pool. Red is the paxillin, the focal adhesions, and white are our mRNA. We could also do puro-PLA, which is a technique that allows us to look at spots of newly synthesized proteins. And for this, you can see the green dots that represents those spots of newly synthesized proteins. We did this for a number of different species. I'm showing a few here, but what we saw across the board is that the untranslated mRNAs, the focal adhesion mRNAs we put in the untranslated pool based on the sequencing data, had significantly less spots of newly synthesized protein near focal adhesions compared to their translated counterparts. Now, all of these assays were done in steady state conditions. So the cells, they're just sitting, chilling in a dish. So we wanted to perturb focal adhesions in a few different ways to understand whether or not we could stimulate translation of these untranslated species. We did 3 different treatments. So we have our controls, of course. We have manganese treatment, which induces integrin activation. We have blebistatin, which decreases actin-myosin contraction, which releases tension on focal adhesions, causing them to fall apart. And that effect is also reversible. So we can do a washout study where then the focal adhesions reform. So when we did this, we again repeated our single molecule FISH assay as well as our PurePLA assay to look at RNA and translation. And so we could quantify the percent of translation near focal adhesions as well as the number of translational spots per cell to see if translation was changing either at focal adhesions or just in general in the cell. This is just one example, but what we found across the board for all the RNA species that we tested is that no, it turns out that whether you treat with manganese, bleb, or the washout, we see no change in the percent translation either near focal adhesions or throughout the cell. So this really reinforces the idea that these mRNAs are not there to make peptides. Now, this is obviously very surprising to us. We were very confused about what was going on. And we chose to take a high throughput approach to better understand the localization patterns of our translated and untranslated RNAs. And perhaps that could give us more information as to their function. Now, unfortunately, with single molecule FISH, as I mentioned, you can only look at one RNA at a time, but we had a collaborator, Stephen Wang, who's in the genetics department at Yale University, and I worked closely with his PhD student, Carol, to do a technique called MIRFISH. MIRFISH, or multiplex error robust fluorescent in situ hybridization, is essentially a technique that allows you to look at multiple mRNA in the same cell at the same time. We can do MIRFISH coupled with a focal adhesion IF to understand the localization patterns of all of our focal adhesion RNAs. We did this assay for both a cell cycle mRNA library control as well as our focal adhesion RNAs. And as you can see, the colored dots represent the different RNA species. White is the focal adhesions. And for the focal adhesion mRNAs, they form these clusters around the majority of the focal adhesions. So we definitely see a difference, again, validating the localization and our sequencing results from before. All right, so now the cool thing that we can do with this data is we can perform mRNA cluster analysis to see if we could define different focal adhesion states simply by their co-localizing mRNA sequences. Okay, so what do I mean by that? So for every focal adhesion, we took what mRNA was co-localized with that focal adhesion and then defined different focal adhesion states simply based on what RNA was nearby. When we did this clustering analysis, we got 4 different focal adhesion states, again, defined simply by the RNA that is nearby or on top of that focal adhesion. We can then go back and look at the focal adhesions themselves and say, okay, is there something unique about the focal adhesions in state 4 versus state 1, for example? And what we found is that there were some focal adhesion size differences as well as circularity between the 2 clusters, particularly between state 1 and state 4. And again, these were only defined based on the RNA that was nearby. What was the most interesting and made us feel a little bit better about our data is that it was the untranslated RNAs that were defining these different clusters. When we look at the feature plots, we can see that their untranslated RNA defined state 1, for example. I have 2 here, track 2 and KIF1C, as well as state 4, again, defined by the untranslated RNA. The translated RNA, however, were spread throughout. They were distributed through all of these different focal adhesions. So to summarize what I've shown you so far, we've identified a population of untranslated mRNAs that localize to focal adhesions. And it's these untranslated mRNAs that can define different focal adhesion states. This is a departure from the central dogma, not what we expected. I was like, oh yeah, they're all going to be translating. We'll follow some targets. It's going to be great. Easy story, wrap it up. But instead, we found all of these untranslated RNAs. And so, of course, our curiosity was piqued and we wanted to follow this idea. So we turned to the literature to think, okay, what does untranslated RNA do within the cell? And to our surprise, there's actually a subset of literature talking about how RNA acts on protein instead of the other way around. And one example of this is in the nucleus, where newly transcribed RNA can help to concentrate protein at its site of function. And this was really interesting to us. So these are examples from the nucleus, not cytoplasmic RNAs, but perhaps that we could be seeing a similar phenomenon with our cytoplasmic RNAs, right? Why are they there? What could they be doing? Could it be to act on protein, as I mentioned, instead of the other way around? So if this is the case, we needed to identify what protein the RNA was interacting with, our focal adhesion mRNA. So to do this, we repeated our focal adhesion isolation assay, this time treating cells for 10 minutes with either RNase A, which degrades the RNA, or cylohexamide, which inhibits translation. We then performed the assay and collected the focal adhesions for proteomics. So the idea behind the RNase A and cylohexamide is that we could differentiate between RNA binding proteins that were responsible for translation versus those that were responsible for simply RNA localization or just interacting with the RNA in general. Doing this technique, we got our top 4 targets I'm showing here that are highly expressed that are then lost with RNase A treatment, but not cylohexamide. We followed G3BP1. I mean, I actually screened all of these, but G3BP1 was our top candidate coming out of the screen because one, it was found to be cytoplasmically expressed, but localized into lamellipodia, these regions here. And that expression went away when we treated cells with RNase A, indicating that RNA is somehow responsible for these localization patterns. We confirmed this biochemically using a pull-down assay. So I pulled down paxillin, and we found that G3BP1 binds to paxillin, but that interaction is dependent on RNA. When we treat cell lysate with RNA, we no longer see that interaction, and G3BP1 is no longer pulled down. Okay, great. So G3BP1, it's at the focal adhesions, it interacts with RNA, but does it interact with our focal adhesion RNA? So for this, we repeated the mirrorfish. So again, this is the technique that allows us to look at multiple mRNA in the same cell at the same time. And with this, we could compare controls versus the G3BP1 knockdown. We used shRNA to knock down G3BP1. And we performed this for both cell cycle mRNA as well as our focal adhesion RNA library. And for our controls, we see a significant shift in the distribution of our focal adhesion mRNA compared to the cell cycle. And that goes away when we treat cells with the shRNA for G3BP1. So when we knocked it down, that distribution, that localization goes away. As a gut check, we also just wanted to look at translation to see if, or transcription to see if we had less overall focal adhesion mRNA, and less than 5% of our focal adhesion mRNA were differentially expressed with G3BP1 loss. So it does seem to be targeted at RNA localization, not necessarily the expression. But what about translation? In the back of my head, I was like, they've got to be translated. Come on, that's what they do. And so we repeated those assays that we like, single molecule fish and puro PLA. So again, looking at the RNA and looking at the newly synthesized protein. And we quantified a few different things. Again, just showing one example, but we see this for multiple focal adhesion mRNA, where when we treat with G3BP1 knockdown or shRNA inducing a knockdown, we see less RNA at the focal adhesion, which is in line with our mirrorfish data. We see no difference in the number of mRNA per cell, which is consistent with our poly-A sequencing data. And we also see no difference in the percent translation, either near focal adhesions or throughout the cell in general. So G3BP1, again, specific to localization, not expression or translation. So how does G3BP1 interact with focal adhesions? For this, we used live cell imaging. I had a Paxillin mCherry reporter and a G3BP1 GFP reporter. We took a number of different videos all in the cell lamellipodia, and I'm showing one example here where we can look at the expression over time of both Paxillin and G3BP1 at a particular adhesion site. What we saw is that on average, G3BP1 expression was delayed by about 45 seconds after the initial nucleation of paxillin protein. But then G3BP1 was expressed pretty much continuously throughout the lifetime of the focal adhesions through disassembly. Naturally, we saw this interaction and we wanted to see, okay, if we knock down G3BP1, what happens to focal adhesion assembly, disassembly? And we saw a dramatic decrease in both the assembly and disassembly rate for the G3BP1 knockdown, which ultimately resulted in a inhibition of cell migration when we performed a wound healing assay compared to the control shRNAs. All right, again, let's summarize. So G3BP1, we know it's present at focal adhesions. It regulates localization, but not expression or translation. And loss of G3BP1 is associated with decreased focal adhesion assembly, disassembly, and migration speed. So we're really starting to put together the pieces of what we think could be going on at focal adhesions. We know focal adhesion mRNA are a piece of the puzzle, and we know now G3BP1 is another piece. Now, G3BP1 was particularly interesting when this came out as our top target because G3BP1 is actually a canonical stress granule protein. So stress granules are formed when cells are under cytotoxic conditions. So these are mainly artificial conditions within the lab that people have studied. So treatments like sodium arsenite, things like that, where the cell completely starts to freak out. There's a bunch of ribosome runoff, untranslated RNAs are hanging out in the cytoplasm, and G3BP1 sucks up all those untranslated RNAs and sort of stores them in these large spherical granules. Now, we don't see these large spherical granules in our cells. We've done IF, we just don't see the same formation, but it got us thinking if G3BP1 could be having another function, because as I mentioned, this is under extreme cytotoxic conditions. Under normal conditions, what might G3BP1 be doing? And so as a note, when RNA and protein interact together, it's called a ribonucleoprotein or an RNP complex. Again, just a fancy word for when RNA and protein interact. And we wanted to understand what RNP complexes might be happening and what's the morphology of those if we're not seeing these really large spherical structures. For this, we used a 2D super resolution approach where we imaged G3BP1 shown in green, Paxillin shown in red, and we could image one RNA at a time. Unfortunately, this technique is not compatible with MIRFISH, so we couldn't look at multiple mRNA. But what we started to see is that G3BP1, first of all, they don't form those really large spherical granules. Instead, they form this heterogeneous population of irregular and branched RNP complexes. So we have the circularity shown here, about 10%, less than 10% are spherical. And there is, a tail to the distribution, but we can see these very non-spherical, very branched, very irregular granule types. And we know that our RNA is integrating into those. I've shown one example here, but we've tested a few others as well. So we know our focal adhesion mRNA is present. And we also know that when we treat cells with RNase A, that granule structure goes away. So the shape, the morphology really is dependent on the presence of RNA, which facilitates G3BP1 organizing in this way. To further understand these RNP complexes, we turn to an in vitro or an in the tube assay, where we take our focal adhesion RNA and we simply mix it together with G3BP1 protein. Our RNAs here are fluorescently labeled, so then we can image them on the confocal. And for all the RNA, the control RNAs I tested, whole cell lysates, BFP, we see the traditional spherical granules form where G3BP1 is organizing in these spheres. We tested 4 of our focal adhesion RNAs, selecting 2 from state 1 and 2 from state 4, as we were still interested in trying to understand what these different RNAs might be doing at these different focal adhesion states. And we had a few findings with this. So first, all of our focal adhesion mRNA complexes were primarily non-spherical, and there were also differences within that. It seemed that one RNA in both states was more elongated and branched. So for example, I have the area and circularity quantified here. TRAC2 and CBLD2 were both much more branched, non-spherical, and much larger compared to KIF1C and IMPAD, which were both present in the same cluster, as you can see here. So why is this morphology important? Like, okay, so what? So they don't make spheres, they make these branched granules. Well, this has been shown very, very recently that the way that these complexes are organized has an impact on their function. And so we wanted to determine what about these RNAs was contributing to this very unique branched structure. We knew even before we had started this assay that our focal adhesion RNAs were unique. They are overall longer. They have a longer 3' UTR compared to non-localized RNA controls. They have a longer coding sequence, which I don't have the graph here, but overall they're just way longer. They also have a higher AU and therefore a lower GC ratio compared to non-localized RNAs. And they also have a higher number of AU domains. And the AU domains were particularly interesting because others have shown that those can contribute potentially to irregular granular morphology. To know which one of these properties was the most important, I took focal adhesion RNA and fractionated them into 500 nucleotide pieces. I did this for the 4 RNA that I showed you before, and we saw that there were 500 nucleotide regions within the RNA that were responsible or that could make these branched elongated morphologies, whereas the rest were all the traditional spherical granules. And so we quantified that here, and we got two very distinct categories of spheric and branched RNAs. So I'm going to be talking about these fragments as spheric or branched RNA fragments. We then looked back at the sequence specificity, but there seemed to be no significant difference. The AU-GC ratio wasn't different. Neither was there an increase in the number of AU domains. And one of our collaborators ended up suggesting, hey, why don't you look at RNA-RNA interactions? I mean, that would make sense. They're making these branch morphologies. And sure enough, when we predicted the free energy of dimerization for our spheric and branched RNA fragments, we saw that the branched fragments overall had a lower free energy, indicating a higher likelihood or more stability for those RNA-RNA interactions. This program is called intra-RNA, and it gives outputs that look something like this, where it actually can predict the regions that are dimerizing. And so I went ahead and mutated some of those regions. So I did 2 different examples of this. And for both of the mutants, when again, we only mutate those dimerization domains, we can drastically reduce the size and increase the circularity of the resulting granules. So based on this, we think that the heterogeneity that we see in cells is dictated by the RNA's ability to dimerize and to interact. Okay, again, let's summarize. So RNA-RNA interactions really determine the RNP complex morphologies. And as I mentioned, the morphology of the RNP complexes is really what determines its function. So let's study its function. This was one of the last experiments that I did, and was a very technically complicated but super fun approach to try. So we utilized something called the MS2 system. So this has been used to visualize RNA in live cells. It's adapted from bacteriophages, where there's MS2 stem loops, which the MS2 stem loops are known to interact with MCP, which is a coat protein that will bind to those stem loops. So what we can do is we can take an RNA of interest, we can attach these MS2 stem loops so that now we have a construct, an RNA construct, that will bind to essentially whatever fusion protein we want, so long as we put the MCP on there. And that's what we did. We took MCP, we fused it to a GFP paxillin. So then that way, what we would have is we would have our RNA of interest being tethered to focal adhesions. The paxillin would incorporate into the focal adhesions. We would be able to see it because of the GFP. And then our mRNA would attach via the MCP coat protein. So we created this system for a few different RNA fragments. I'm going to show you 2 here today. So TRAC, these came from TRAC, either a spherical TRAC fragment or a branched fragment. Okay, so with this, we use single molecule FISH to validate that we got these clusters and an increased number of our spheric or branched RNA at focal adhesions compared to just the endogenous. So we do still have the endogenous, but we now have this increase because of the way that the tethering assay works. The first thing that we studied with this technique was paxillin protein mobility. So because the paxillin is fluorescently labeled, we can go in and we can frap the fluorescent recovery after photobleaching. We can frap the focal adhesions and see what happens. We did this for control spheric and branched RNA. And what we saw is that with the branched RNA in particular, we saw a reduced % recovery rate compared to both the control and the spheric RNA. So it seems that there's something about the protein mobility simply by expressing RNA at the focal adhesions that's limiting its ability to get into the focal adhesion after frap. We also looked at focal adhesion assembly, disassembly rate, and focal adhesion lifetime. So first, I'm going to walk you through the spheric data. So what we found is for the spheric RNA, so again, we're just tethering a spheric RNA to focal adhesions. We can increase the assembly rate, increase the disassembly rate, which ultimately decreases the focal adhesion lifetime. Conversely, for the branched RNA, we still increase the assembly rate, but we dramatically decrease the disassembly rate compared to both controls and spheric RNAs, which ultimately leads to an increase in the focal adhesion lifetime. Looking at migration, we think that the data is consistent because for the spheric RNA, which has these very quick focal adhesion or very short focal adhesion lifetimes, we see significantly more migration over the course of 12 hours, whereas the branched RNA, which have longer adhesion lifetimes, have limited cell migration. So on top of this, when we treat cells with G3BP1 shRNA, all of that goes away. So it seems like it's the combination of the effect of local RNA as well as G3BP1 that controls the way that these RNP granules somehow change the biophysical properties around the adhesions that leads to these alterations in migration. So to put everything together, again, these mRNA fragments, so again, we're just talking mRNA fragments with distinct RNA-RNA interaction properties that can be tethered to different focal adhesions to regulate cell migration. And that, of course, as I showed, is dependent on G3BP1. Our proposed model at the moment is that essentially cytosolic mRNA regulates protein complex organization in a non-coding function. So these are not being made into protein. They're simply changing the biophysical milieu around the focal adhesions themselves. And it's really the dimerization property that could differentially drive the way that these different protein complexes function. So we propose that really it's the untranslated mRNAs, as I mentioned, with the different dimerization properties that could control cell migration speed. So what are we doing now? Well, this was a ton of work. We're trying to get the paper published, but just closing out a few minor things. One is tethering the endogenous. I had some issues with the cloning, and so we're trying to understand the difference between the state 1 and state 4 focal adhesion mRNA. The fragments behaved the same way. So when I did the fragments from both state 1 or state 4 RNA, it didn't really make a difference. We saw the same results. So what happens if we tether the endogenous? We overexpress a state 1 focal adhesion mRNA at all focal adhesions, not just the ones that they're endogenously localized to. So that's the last sort of on the ground experiment that we're doing. I'm also starting to work in zebrafish. So our lab does both zebrafish and cell culture as model systems. So right now, in collaboration with one of the graduate students in the lab, Peter, he and I are working to generate G3BP1 mutants. And our initial data suggests that the focal adhesion G3BP1 complex also regulates cell migration in the vascular system. So we want to understand more broadly in 3D tissues and organs, but the very first phenotype that we noticed was in what are called the intersegmental vessels, or the ISVs. So I've shown the diagram of the vascular system here in zebrafish. And what we saw, we have videos of this as well, but we saw first a delayed migration, and then also an issue with the G3BP1 mutant in terms of cell-cell contacts. They would either detach from the bottom vasculature or never be able to reach or attach to the top vasculature. So we think that this isn't something that is simply in a cell culture system, but also in organisms as well. And in terms of the big picture, how do we exploit this phenomenon for RNA therapeutics? We want to be able to utilize what we found in order to not change protein, not change RNA expression, but simply to be able to express non-coding RNAs to regulate the way that cells behave. And so with that, I'd like to acknowledge the Nicole Lab. This work would not be possible without the help of Lauren and Peter, both graduate students within the lab, as well as our fantastic collaborators, Stephen Wang, who helped all the mirrorfish, his graduate student Carol, as well as Christine Mayer at Sloan Kettering, who helped with a lot of the RNP data. And of course, I'd like to thank my funding sources. So thank you very much for the opportunity, and I'm happy to take any questions.
Very nice. Very nice work. Thank you.
Great. Thank you so much.
Yeah. All right, we'll take questions. You can unmute yourself if you have questions, and or use the chat. While they're doing that, I have a question.
Mm-hmm.
So as you just mentioned here at the end, again, very nice work. So when you start to get into a more complex system like the zebrafish,
Mm-hmm.
what are you going to be looking for? So are you able to use similar techniques, or are you going to be limited, maybe just looking at the G3BP1 effects?
Yes.
So are you looking at RNA? Go ahead.
Yeah, no, definitely. Fantastic question. So we started with the G3BP1. It was sort of the easiest thing we could do. We routinely use CRISPR-Cas9 in the lab to knock stuff down. We wanted to see if just knocking down G3BP1 had an effect. But as you're absolutely right, we want something that's more targeted. G3BP1 can, as we see, have effects under stress. So we don't know what that looks like within the cell. So what we want to do is to utilize the same tethering system that we used within the cell culture and use that within the zebrafish. So we know that the MS2 system works in zebrafish. So it's just a matter of testing out that fusion protein and seeing whether or not we can do the MCP, GFP, paxillin in the zebrafish, but there's no reason why it shouldn't work.
Hi, Liana. This is Yale Goldman. Can you hear me?
Yes, I can hear you.
G3BP1 is a typical stress granule assembly factor, a marker for stress granules, and then it has all these other functions like splitting RNA DNA duplexes and even involved in RAS signaling. So with all these different putative functions, do you know how it works in your association with the focal adhesions? Are any of those functions related? Or if not, what is it doing?
Yeah, that's a great question. So G3BP1 really is, as you mentioned, sort of ubiquitous. It does a lot of different things with interacting with untranslated RNA and just RNA in general. So we do have a few ideas of how we can potentially address that. So at the moment, we do know that the function of the RNA, when we tether the RNA, is dependent on G3BP1. Now, how that's having an effect, we don't necessarily know. We have a few different G3BP1 mutants that we want to test within the lab. We also have talked about doing a paxillin. We know paxillin and G3BP1 interact. So if we can try to mutate that interaction to really just change G3BP1's function at focal adhesions, we think that those types of assays could help address the question that you're asking.
Okay, thanks.
Questions? Any in the chat? It's a silent bunch today. Let's see.
I have a question, maybe a little far out there. So first of all, very beautiful work. So I'm thinking about the function of filopodia. And so your work focused mostly on the focal adhesion component of filopodia, but they also play like a significant role in other aspects of probing their environment, like looking for other signals. And I'm wondering if what you would expect based on your data, if they would play a role in kind of collecting environmental data in any way.
Yeah, I think that's definitely possible. So one thing that we noticed with the mirrorfish, as well as some of the single molecule fish data, is that the RNAs, we also saw them in the filopodia as well. But those were actually the ones that seemed to be more translated. So I think we've studied the untranslated RNA because of the high number that we found. But I think that there's a lot of stories that we really need to dig into when it comes to the translated RNA, because we have especially a lot of really important proteins like filament and talon that are all translated, and those are the ones that seem to be in the Filopodia. So I do think that that's probably more likely an explanation compared to the untranslated RNAs.
Oh yeah, that makes sense. Yeah.
Yeah.
Okay, there's chat. Jose, do you want to just unmute yourself? Let me read. I can't read. Hold on. Okay, his question is, have you noticed any changes in mRNA due to different ECM coding like fibronectin or collagen 1? I guess how did you conduct these experiments, and if, you know, would you expect to see changes, or did you look at that?
Yes. So first of all these experiments were done on fibronectin. That was what we had started because of actually the microRNA study that I had done when I first joined the lab. We were just doing everything on fibronectin, but I did test fibronectin, laminin, collagen type 4, and collagen type 1. And we didn't see any differences in RNA localization per se, but we did see different effects of the G3BP1 knockdown depending on the ECM. So that definitely needs to have further investigation. But I think that there's some interesting ideas there that could be probed.
Okay. Any other questions? Okay, Liana, we thank you today for a very interesting talk.